Our next speaker is Elyse Bolterstein from Northeastern Illinois University. Elyse, can you share? I can see your slides.
Perfect.
Take it away.
Okay, thank you everyone for joining us today, and thank you to our wonderful chairs for giving me the opportunity to talk about my cure. And I'm also very fortunate to be the spokesperson of this collaborative cure that I work with my colleagues Catherine Cole, Eric Stockergen, and Christina Swanson in looking at mapping and characterizing mutagen sensitive genes in PUIs. So many of us are here because we already understand and love cures. Casey gave us a really nice introduction to it. Of course, in a cure, you're asking your students in the classroom to answer research questions in which the answers are unknown to the faculty and the students, and they're of interest to the general scientific community, which is giving everybody a really nice, authentic research experience built into the classroom. As we know, there are many benefits to running cures, both for faculty and students. And the one that really rang very true to our group is the idea of increasing research accessibility for our students. So traditionally, when we think about research experiences, we tend to think about more intensive research experiences where you might spend a summer, you might spend a few semesters in a faculty's lab. And when we're thinking about this in our particular institutions, we all teach at schools that have a large percentage of students that are traditionally underrepresented in science, such as students who Generation American or students who identify as minorities. And at my home institution, Northeastern Illinois University, we're classified as a minority serving institute. And I know a lot of my students have personal and financial constraints in terms of their responsibilities at home or to work that may prevent them from being able to do a more intensive research opportunity. We also, on the flip side, can sometimes have limitations in faculty resources in terms of time and funding in order to have big enough labs. So by running our cures, especially in core undergraduate classes, it's a way to bring research to many students so that they can have the benefits of participating in a more independent research experience. So before I talk a little bit more about a cure, I just want to talk about classroom logistics. All of us teach small lab courses in our schools with about 15 to 24 students per class. We typically meet for lab once a week, having a structure of one lecture, one lab. Our students, depending on our project, are divided into teams of 3 to 4, and they design the research project and they execute it and then they present. Now, depending on the project and the course, this research is carried out in 1 to 3 month modules, and their overall cure determines about 10 to 15% of their grade. So our cure starts with mapping the genes. And to back up and talk about the must genes, the mutagen sensitive genes were first determined in the late 70s, early 80s to be sensitive to EMS, or sorry, they were determined by EMS screen to be sensitive to different mutagens, including MMS. However, in the last 40 years, only 14 of the 58 genes have been mapped, giving us a really ripe opportunity to study these genes. So our first candidate that was chosen is MUS109. It's an X chromosome gene. We were fortunate enough to have 3 alleles readily available to us through Bloomington to work with. And there was also evidence that it's sensitive to several DNA damaging agents, showing that it is important in different kinds of repair. So our deletion mapping process started at Winthrop University in Catherine Cole's genetic classes, and she used complementation crosses in order to narrow down where MUS109 is on the X chromosome. And this worked out really well because, as many of us teach genetics know, complementation crossing is part of a normal genetics curriculum, so it fit in really nicely with the class. So briefly, what Catherine did is she used 4 different MUS109 area deficiency flies and crossed them with the MUS109 alleles. And the students could easily determine who was mutant and who was not mutant after MMS treatment by the bar-eyed phenotype. And serendipitously, they found that all 4 deficiency flies failed to complement 9 alleles and brought them to a smallish region of the genome that only had 9 genes there. And of those genes, one of them was projected to have 5' flap endonuclease activity and was also projected to be orthologous to DNA2. So this was our DNA repair candidate. Now that's where they did all the mapping in genetics class, and Catherine's students then took over in her own research lab to finish the nucleotide sequencing that way. Now, after Catherine worked on the mapping, she went on to do other genes, and the rest of us started working on their characterization. So in my genetics class at Northeastern Illinois, the students looked at mutagen sensitivity in the larvae, where they were able to pick which mutagen they wanted to treat their larvae with and predict what's going to happen. And I have two students who are presenting posters that are a spinoff from this project next week. In Eric Stoffergen's class at Lewis Clark State College, his genetic students looked at fertility and were able to confirm that MUS109 mutant females are sterile. And in Christina Swanson developmental biology class at Arcadia University, they looked at ovarian development and found that the nurse cells in particular looked abnormal. All three of these projects are being followed up on in our own research labs so that we can more effectively look at that characterization. Now, we were lucky enough that every semester, at least two of us were teaching a class at the same time. So this meant that the students could share their research, not only with each other in the classroom, but also across the different institutes. So what we did is design a virtual poster session. When we first did it, it was very new to us. We didn't know what Zoom was. Of course, we didn't know that's what our lives would be like now. But the students were able to record their presentation in class and engage their own peers with a live question and answer session. Then we put the recordings up on a Common Course website so that all students in all courses could talk to each other and comment on each other's research asynchronously. This was awesome. I was so impressed with the questions and answers that my students were giving. And we also were able to stagger the question and answer times between classes so that they always had something to do. This also gave the student exposure to different experiments so that they could better understand that to put together an idea or research or paper, it actually comes from multiple sources and multiple experiments, not just one. So being that we are all educators, we were really interested in learning gains and what happened in the classroom. So we used a survey to look at student learning gains and also to evaluate our cure in terms of meeting criteria that have been set forth by others in the cure literature, such as Aaron Dolan. So we found that students gained a lot in research skills, which is good. That's what we were hoping. And especially in experimental design, communication with each other, and also collaboration with their groups, with different teams in the classroom and across the universities. They also gained skills in data analysis, especially when they were working on statistics, which was great. What I think was the most important finding we found in gains is that they reported that they were more confident in talking about, communicating, and thinking like they are scientists. And this is really important to us. Like I said before, most of us teach students who are traditionally unrepresented in sciences. And what's key in them going forward is to be able to see themselves as scientists and understand what it's like to do real science. So enabling them to have this high confidence was very important to us. We also found that we met cure expectations for collaboration and discovery, which is great. Of course, everything is a work in progress, and especially as scientists and educators, we're always trying to improve upon what we have. One thing that was a little bit of a snag is that even though our core courses were great in terms of involving a lot of students, they had high content expectations, which meant that the students felt a little crunched doing their research project in some cases. For example, I teach genetics. My project was on DNA repair. For those of you who taught genetics before, you know that it's kind of a big lift to go from Mendelian genetics on day one to DNA repair in time to really fully understand the cure. And even with our scaffolding and unveiling of different parts, some of the students still felt a little overly challenged in that respect. Another cure criterion that's important to think about is to be able to give the students time to revise and redirect their projects if need be based on their data and circumstances and more they learn in the class. And again, because of that high content expectation and how we were fitting the cure into our classes, we didn't really have time for that kind of thoughtful revision. And the students wish that they could have another few weeks to do another experiment, which is something that we're going to consider in future iterations. Lastly, like I mentioned before, all of our courses were run where they would meet in lab once a week. My course was a little lucky because we got to meet twice a week in the same room, so I could use lecture days for lab. But the students had limited access to the research lab, which meant that everything we had to do either had to happen within the span of a 3-hour lab, or would happen outside, done by ourselves or prep staff or help, when the students felt like they lost touch with that hands-on experience when we would just tell them what would happen. So we're thinking about ways to redesign experiments, or perhaps have students have better access where they can come in at different times. Now, to meet these concerns and go forward, we were kind of considering the types of experiments we're running and the types of courses we're running them in. We think that mapping in the general genetics class is a perfect fit in terms of the core curriculum and how students can understand and connect with the experiment. Like I mentioned, doing the characterization, depending on the course and the experiment, might have been a little crunchy for the students. So we're thinking about creating upper level courses in which the students can kind of come in with a higher content or higher knowledge base, so we can get into the cure a little easier and they can feel more comfortable. The students love being able to present to each other. We're also looking for more interaction in terms of sharing what they have with the scientific community, in terms of timely conferences that come up at the right time during our classes, maybe engaging them in doing mini publications or other ways to get their research out there before we are able to do it all as a group. And lastly, we've been doing this together for a few years now, and we're interested in growing our cure network. We kind of have a more the merrier philosophy. And if we can have more people do this, we can answer questions more quickly and come up with ideas, as you know, that we probably wouldn't have ever thought about before. So with that, I'd like to acknowledge some of the folks that have been working on this cure. Catherine's lab students, Chandani Mitchell and Jordan Deloach, did the nucleotide sequencing in her lab. Those of you who do cures know that it takes a village, or at least a lot of work, in order to get everything done. So we'd like to thank all of our lab managers, prep teams, student assistants, in terms of being able to help get all the flies to the students in time. We've had 10 classes participate in the cure so far, about 200 students. And so they've all been great. And lastly, we like to thank the Ciliate Genomics Consortium for their funding, advice, resources, everything they've been able to give us in terms of launching our own cure. And included a picture of most of the team the last time we saw each other in person in 2018 at the Philly Fly meeting. And an encouragement that please get in contact with us. We'd love to talk to you. And if you'd like to see more information on the project, including some of the both the research data as well as the learning gains data, you can check out this website. So with that, thank you everybody, and I'm happy to answer any questions.
Thank you very much, Elyse, for that great description of your cure. So there are some questions in the Q&A box. So did you have students use mutagens? And if so, what kind of precautions did you have to have them work with?
That is an excellent question. We had the students decide what mutagens they were going to use, work out all the concentration, but I was the one who actually dosed them with mutagens just because of the safety concerns.
Okay, awesome. So we have a couple questions about sort of virtual environments. So could you continue this cure in a virtual learning environment, and how could that be altered if necessary?
Yeah, that is an excellent question to which we haven't really come up with an effective answer yet. When everything went remote, we were finishing up one semester, and it was at a time where we could just finish up experiments, or they were already done and we could present. This past year, everybody's taken a step back from the cure just because of thinking of how this would work. In the future, something we might want to do is have the mapping staggered, so that if we have one class do the mapping, or if mapping is done by summer students, we could have the sequencing done by the students in the actual class. So we have ideas, but we haven't actually tackled it yet. And I'll throw it out there again. If anybody else has ideas about how this could go into a great remote project, I'd love.
So you should look, Elyse, in the question and answer, because there are some people who are, volunteering to listen to presentations, right? So now that things are going to be on Zoom, right? One person was wondering if you could have different institutions be involved and have folks come and watch presentations and that sort of thing. So that is something to consider. And Brian Oliver is also plugging micropublication biology, which might be a good home for some of these results. And I know there's a workshop with micropublication biology next week to learn more about what they do. So will the students be authors of papers that include their research?
So that's an excellent question. I see that the follow-up is it a problem that this will involve a lot of authors? And I'd say no, just based on Sally Algin's work, where I think there's one paper with over 1,000 authors on it. In terms of student authorship, that's something that we're not there yet in the cure. I would say that we're going to have to come up with the right criteria for how far students are going to take it in terms of reading over the drafts and being part of the authorship. Again, we're kind of at the earlier stage of our cure and we haven't yet published. So that's something I'd love to talk to more of you who are more seasoned in the cure environment to kind of help dictate how we might make those decisions.
All right, so for time, we need to move on, but Elyse, you can answer the remaining questions in the Q&A. All right, so on to our next speaker.
Thank you, everybody.